Welcome to The Explainer. Today, we're stepping into the shoes of molecular detectives to investigate one of the most frustrating and honestly, deadly medical mysteries in modern oncology. I mean, imagine this. You've got a highly effective weapon against a terrible disease. You use it, and it only works on half of the target. The rest of it just sits there, completely unharmed, continuing to grow. Well, that is the exact puzzle researchers are trying to solve in a groundbreaking new cancer cell study, and we're going to break down exactly how they're doing it.
Okay, let's dive right into this by looking at the clinical reality we're facing. When patients with non-small cell lung cancer get immunotherapy, the whole idea is to unleash the patient's own immune system to attack and destroy the tumor. Sounds great, right? But very often, doctors see this incredibly maddening result. Parts of the tumor shrink and die, but other parts just totally survive the attack, and you have to ask yourself, why? Why does the exact same immune system inside the exact same patient completely destroy one side of a tumor but just ignore the other?
So in the clinic, they call this a partial response. A partial response basically means the immunotherapy only wipes out a portion of the tumor. And that means the surviving resistant cancer cells get to persist, they mutate, and eventually they cause the disease to progress. Now, this matters immensely to patients because it reveals something terrifying but absolutely crucial. A tumor isn't just one single uniform thing. It's more like a highly diverse gang of cellular suspects, all living in the exact same neighborhood. And some of those suspects, they know exactly how to hide from the cops.
Section 1. Isolating the tumor suspects. 81. Now that number is wild when you really think about the sheer scale of resolution these scientists achieved. Using data from the TRACERx lung cancer cohort, they didn't just look at whole intact tumors. No, they acted like microscopic detectives. Across just 3 non-small cell lung cancer patients, they isolated 11 different spatial regions of the tumors, and from those, they extracted 81 distinct clonal sublines. Essentially, they grew 81 genetically distinct mini-tumors from just 3 people. This let them interrogate every specific family of cancer cells completely individually.
So here's how you actually set up a biological police lineup. Step 1. They extracted these multi-region organoids, which are basically 3D microtissues grown in a dish that mimic the actual real-life tumor. Step 2. They isolated the individual clonal sublines, meticulously separating out all those different gangs of cancer cells. And step 3, well, this is where the magic happens. They co-cultured these isolated cancer clones right alongside immune cells. For 2 patients, they used the patient's own T cells, and for the third, they used donor natural killer, or NK cells. They literally put the immune cells in a dish with the specific cancer clones, pulled up a chair, and watched them fight it out.
This whole highly sophisticated setup was designed to answer one fundamental question. Do different subclones within the same tumor have different intrinsic abilities to evade immune attack? Now, the absolutely key word there is intrinsic. By taking the cancer cells totally out of the human body and testing them under the exact same controlled culture conditions in a dish, the researchers completely stripped away all those local neighborhood factors, like physical barriers or a lack of oxygen. If a cancer clone survived the fight in the dish, it meant the evasion tactic was baked right into that cell's own biology.
Which brings us to Section 2, the intrinsic evasion discovery.
So, here's the huge reveal. They actually found definitive proof of subclonal immune evasion. When they mapped out the genetics, looking closely at the cell's complete DNA blueprints and their active genes, they found that the immune-evading clones, the tough ones that survived the dish fight, and the non-evading clones mapped perfectly to entirely different evolutionary lineages. These hot clones that died easily and the cold clones that survived had literally evolved differently from one another inside the exact same patient. Evasion isn't just random luck, it is a heritable intrinsic property.
And get this, they don't even use the same tricks. In patient 1, the evasion was antigen-dependent. The cancer cells essentially hid their identifying markers, so the T cells just couldn't see them. It's like using a fake ID. But patient 2, completely different story. Their evasion was antigen-independent. The immune cells could see the cancer perfectly fine, but the cancer cells just resisted the attack anyway, almost like they were wearing a biological bulletproof vest. And patient 3 showed a totally different vulnerability entirely, with huge heterogeneity in how natural killer cells recognized them, which was tied to their MHC1 expression, acting kind of like a cellular ID card. Every single tumor gang uses completely different hustle tactics to survive. Moving on to Section 3. Descriptive versus functional oncology. Now, this slide brilliantly illustrates a core shift in how we're studying cancer today. Previous studies? They were mostly descriptive. Researchers would look at DNA or RNA data or maybe static spatial images and kind of infer or guess that immune escape was happening based on the mutations they saw. But this study is functional. They didn't just guess, they directly tested whether these living tumor clones could actually activate immune cells in a dish. We are no longer just staring at a blueprint, we're plugging it in and watching the machine work in real time.
And the researchers proved exactly why this functional approach is so necessary. They found that genomic and transcriptomic data alone are fundamentally insufficient to reliably identify which clones will evade the immune system. If they had only used descriptive DNA and RNA data without actually making the cells duke it out in the dish, they would have completely missed the functionally immune-evasive clones. This was especially true in cases like patient 2, where the standard markers just didn't explain the resistance at all.
Section 4. Study limitations and caveats.
Okay, we have to be totally objective here and talk about the limitations. First off, the sample size is incredibly small. They only got 3 functionally studied patients out of 42 attempted. Growing these complex organoids is genuinely really hard. Second, the model is pretty reductionist. Real tumors have complex physical barriers, hypoxia, and supportive cells like fibroblasts that this dish-based model just doesn't capture. Third, they had to use donor NK cells for patient 3, rather than the patient's own T cells. And finally, these patients were treatment naive, meaning we just don't know yet if these exact dynamics hold true for tumors that have already survived heavy rounds of chemotherapy.
There's also one specific data caveat I should quickly note. The authors actually had to issue a correction because some of the RNA sequencing treatment labels for patient 1 got accidentally switched in the initial analysis. Hey, mistakes happen. Now, this meant about 7% of the initially significant genes lost their significance, which required some of the pathway results to be revised. Actually, wait, let me clarify why this isn't a deal breaker. The main conclusions of the paper absolutely still stand. Why? Because the core discovery relies entirely on those live functional cell assays we've been talking about, not just the RNA data.
Section 5. The future of cancer therapy. So, where do we go from here? Well, this builds a massive foundation for the future of oncology, and the next steps are pretty clear. We need to test this platform on much larger groups of patients across all different cancer stages. We need longitudinal sampling. I mean, imagine tracking these exact evasive clones over time, comparing the original primary tumor to a metastasis that pops up way later after therapy, just to see if the evasive clones are the ones that actually seeded the spread. And we definitely need to make those dish models more complex by adding in macrophages and fibroblasts to better recreate the true chaos of the human body.
Plus, we've got some incredibly cool unsolved mysteries to crack. Remember patient 2, the one with the biological bulletproof vest? Standard DNA markers couldn't explain how those cells were fighting off the immune system. So to figure that out, future researchers are going to have to bring out the big guns. They'll need advanced tools like CRISPR gene editing screens, proteomics, and single-cell perturbation assays to finally find that invisible shield patient 2's cancer cells are using.
But bringing it all back to the patient, right? Because this right here is the ultimate clinical goal. The whole reason we want to map out every single tactic these different cancer subclones use is so we can design highly targeted combination therapies. Think about it. If we know a tumor has one gang of cells that hides its antigens and another gang that uses a bulletproof vest, we can give the patient a specific drug cocktail that targets both mechanisms at the exact same time. The goal is to stop partial responses entirely, wipe out the whole heterogeneous tumor, and achieve complete remission.
Which leaves us with this final, really profound question to consider today. Using these functional dish models, will we one day be able to predict exactly which subclones will resist therapy before a patient even begins their treatment? Imagine taking a biopsy, testing the clones in a lab, and knowing exactly what customized cocktail of drugs to give them on day 1. That is the incredible future this research is pointing toward. Thanks so much for joining me on this explainer. Keep asking those big questions and never stop staying curious about the science that is changing our world.